Now it's my pleasure to introduce Nigel French. Nigel French is interested in molecular epidemiology and pathogen evolution, and he works at Massey University. He's currently director of the New Zealand Food Safety Science and Research Centre, and also director of the Infectious Disease Research Centre. Thank you.
Thanks very much, Tom. Thanks very much to the organisers for giving me the opportunity to come and talk to you today. So I'm going to talk about food safety, a little bit about water quality, a bit about public health, and a little bit about biosecurity, but as it relates particularly to transfer of pathogens between livestock sectors. So this is just an outline of what I'm going to cover, what's changed, what is the revolution, and how we're applying those technologies in a number of key areas that I mentioned there. And then I'm going to give some examples going back historically to 10, 15 years of how we're looking at a number of pathogens that have affected public health, but also had an impact on our livestock industries and wildlife as well. So I'm going to talk a bit about Salmonella, a bit about one strain of E. coli, the sugar toxin producing E. coli, and then talk a bit about Campylobacter. Two pictures that we have up there. The black and white one is Campylobacter. That's a wiggly bacteria with a flagella at both ends that wiggles its way through your gut mucus. And the other one is E. coli. So what's changed? The revolution in whole genome sequencing means that we can now sequence whole genomes rapidly and relatively cheaply. So some of the techniques that we've been using historically are actually now more expensive than using whole genome sequencing. And we can now combine some of the newer sequencing technologies to get really good, high-quality information from genomes. And I'm specifically interested in microbial genomes. But the other revolution that's happened is that we can now use that information much more effectively to infer and answer questions related to public health, to transmission, to the source of human infection. And many of the tools that have been developed have actually been developed by New Zealand scientists, and many of these were scientists within the Alan Wilson Centre. And there's a few examples along the right-hand side of key software tools, which are almost the global standard for a lot of these analyses, and many of these all of the ones on the right were developed by New Zealand scientists. So New Zealand's a fascinating place to study this whole issue because of a number of reasons. We do have a relatively low diversity of pathogens, including a number of pathogens which are fortunately not present in New Zealand. These include pathogens such as Q fever, Brucella abortus, a number of key pathogens of importance to livestock health and to human health are just simply absent from here. We also have a number of very unique strain types as well. We seem to have sampled historically from what's globally circulating and allowed them to evolve in our relatively isolated ecosystem over a number of years. So the population structure of these pathogens has been shaped by a whole number of different factors that I mentioned there. So movement into the country historically through immigration of people and live animals, the relative isolation, and the limited subsequent importation of animals. And then we have bottlenecking and demographic expansion, which I'll cover a little bit later on. So the first example I'm going to talk about is a Salmonella example. Salmonella is common throughout the world, still a very common cause of food and waterborne infections globally. And in New Zealand, we have around about 1,000 cases per year of Salmonella. And these Salmonella cases are usually dominated by one or two strains. And in 1998, we saw the emergence of a new strain of Salmonella, which is called DT160. And the unusual thing about this DT160 case was it caused a large outbreak of Salmonella in people, which was sustained. It was the dominant strain for at least 10 years. But it also caused a major die-off of small birds initially around the Christchurch area. So any of you from Christchurch may remember seeing lots of sparrows dead and on bird tables, and it was quite a marked death and die-off of these small birds. Now that subsequently transmitted then into livestock populations, so it got into farmed animals and into other wild birds. And then it progressed very rapidly from its focus in Christchurch to cover the whole country, and I'll show you a movie in a second of how that happened. Unfortunately, we have a very good isolate collection, which is stored at ESR, at the Enteric Reference Laboratory. So we're able to use that resource to then look historically to see what happened and what the implications are for public health and also for potentially for biosecurity. So the graph on the right shows the epidemic in I'm not sure whether I've got a pointer. So this is the epidemic of DT160. It replaced the previous strain, and there was a big die-off, as I said, simultaneously in wild birds as well. So the next map I'm going to show is just an example of 3 different strains of Salmonella. The one that I'm going to be mainly focusing on is the one that was sparrow-associated initially, and you can see in green here the first few human cases in the Christchurch area, one down here in Invercargill, but no other cases around in North Island or the rest of South Island. And we can contrast this with the strain which was associated with mainly sheep in blue in the southern part of South Island, and another strain in purple which was associated with cattle, particularly in North Island. And every second is about 2 months, so when I play it, if it works, you'll see how rapidly the epidemic then spread. You can see already within a couple of seconds, so this is within the first few months of moving out from its focus in Christchurch, it was right at the top of Northland and basically throughout the country. And this was then sustained for a period of about 10 years. So what we've done is we're interested in how this evolved over time and what the potential implications were for public health as well. So one of our PhD students, Sam Bloomfield, working with ESR and the Enteric Reference Laboratory, genome sequenced 109 isolates from that collection. And these were from a range of different sources, so including the first human case in 1998, and a number of other ones from poultry, from wild birds, and from cattle. And what you can see is that the human case, in terms of its phylogenetic relationship, so isolates that are further apart on this tree are more distantly related to ones that are closer together. And you can see the first human case is right at the center of the phylogeny. And then further, as you move through time, you can see radiating out, and the more recent isolates are more distant in the tree. And this is a classic example of a radiating evolution of a pathogen from a point source, an introduction, and you can just plot that graphically. You can see it's almost a monotonic increase in the diversity over time. So this is an example of a pathogen that probably came in as a point source into Christchurch and then radiated out over time. And what we can also do using these evolutionary modeling techniques is we can also show how the bacterial population changed over that time. And what we saw was a very marked increase, probably as it was amplifying in wild birds and then crossing over into livestock, which coincided with the human outbreak as well. So we saw this huge increase in the bacterial population. The other thing that the genetics showed was that there was a lot of transmission ongoing right the way throughout the epidemic. It wasn't just wild birds to people, it was wild birds to animals, animals to people, and probably even people back to animals as well. So there was inter-host transmission happening right the way throughout the epidemic. So how do sparrows infect people? Handling of dead birds was identified as a risk factor. We also know it transferred into food animals as well, but you also see quite a lot of this kind of thing happening as well, which is almost certainly an important route for transmission as well. You can see contamination of the general environment, particularly food preparation and food eating areas could be a common source of infection. Roof water and playgrounds being other potential sources as well. The footnote to the Salmonella DT160 story was that we were approached by the Melbourne Diagnostics Unit. They'd also had a sustained outbreak of this same pathogen in Australia, first acquired in 2008, so a bit later than the New Zealand outbreak, also associated with sparrows and also predominantly in Tasmania. And that was the phylogeny of the Australian strains. Now, in addition to that, they also had a cluster of cases which had happened previously in 2003. So we then shared the data that we had from New Zealand, and what you could see was that that cluster of cases fell right within the New Zealand group. So there was some evidence of a link between what was happening in New Zealand at the time those clusters happened. And it turned out when you looked at the epidemiology, this was a cruise ship, so these were passengers on a cruise ship that arrived from Auckland to Sydney, so it was possibly a transmission event that happened directly as a result of humans moving between the two countries. The next pathogen I'm going to talk about briefly is sugar toxin producing E. coli. Now, the first case of E. coli happened in New Zealand in 1993, in a person this is. Now, the reason why I was so concerned about this strain of E. coli is it's important because it's declared an adulterant of beef products or meat products, so it's important for our food industry when we're exporting meat overseas. But we're also getting an increasing number of cases each year of bloody diarrhea, and it's also associated with kidney failure and death in people as well in New Zealand. And it's particularly preschool children. You can see that there's a peak in the number of cases in every year. This is 2016. About 400 cases were notified last year. And you may have seen from the movie that a lot of these cases are also associated with high-density dairy farming areas. So areas such as the Waikato, Taranaki, and also South Canterbury are common areas where you see these outbreaks. So we applied genome sequencing. We were interested in asking a couple of questions. When did 0157, this particular strain of STEC, come into New Zealand? And could we use genome sequencing of historical isolates to determine what happened? And what we could show was that we had 3 different clusters of 0157. And when you timed the common ancestor using historical isolates going back over a 10-year period, you can get a rough estimate of when they're likely to have entered the country. And these strains were dated around about 1970 through to the end of the 1990s. So this was key because this means that they're relatively recent introductions. They weren't introduced 100 or 200 years ago. They're relatively recent. If you time all of the groups together, it comes to around 1848, which turns out to be a good estimate of when the globally circulating E. coli population of STEC converged over time. So again, we seem to have sampled from the global population relatively recently in New Zealand. And this does happen to coincide with when there were increases in imports of live cattle into New Zealand, so we can hypothesize that this importation of animals around about this time may have been when E. coli 0157 was introduced into the country. Other things that might support that hypothesis is that we also see that the bacterial population of that strain of E. coli also increased from about mid-1980s onwards. This is also when the number of dairy cows also increased, so dairy cattle and cattle in particular being the major hosts for this particular pathogen. And we can also see an increase in stocking density, a decrease in herd size, an increase in the number of herds as well. And we know that all of these things can be associated with ongoing transmission. So one scenario that we're suggesting might be possible is the 1970s onwards introductions of live animals might have brought this new pathogen into the country, or different strains of this new pathogen, and the increase in the dairy industry may well have amplified the pathogen, and then subsequently we started to see human cases occurring around about the early 1990s. So the last pathogen I want just to focus on is Campylobacter, and this is obviously one that's very commonly in the news. It's associated with poultry, and last year it was also associated with a major waterborne outbreak in Havelock North. I'm going to be talking very briefly about those things, but first of all, just to remind you that we had an increasing incidence of Campylobacter infections in people up to 2006. And this was a growing epidemic over many, many years, and it really was a lot worse in New Zealand than anywhere else in the world. Although we have good reporting systems, we're not that good that we're orders of magnitude better than everywhere else in the world, so I think it really was a growing epidemic that was happening over that time period. Michael Baker from Wellington School of Medicine published this paper, which was the trigger for a lot of activity to try and determine what was the real cause and source of Campylobacter in New Zealand. There was a lot of controversy around the time. Was it chicken, as Michael was proposing? Was it cattle? Was it environmental water? There was a lot of speculation around at that time. We set up the Manawa 2 as a sentinel site. We sampled from food, from humans, human cases, from water, from farms in catchment areas, and we did typing on those, and we used a combination of those plus modeling approaches to determine what the likely source of those Campylobacter infections were in humans. And in the early days, we had a 7-gene scheme, so this is before whole genome sequencing was really cheap and available. And we were able to determine that this was the kind of population structure that we saw. A lot of isolates such as this one, which I'm going to come back to, sequence type 474, which was the predominant strain in humans in those early years, but was found almost exclusively in one particular poultry supply. So this was in those early years that was very, very strongly associated with poultry. Now, we combine that information with modeling, and these are the models that were being developed at the time based on evolution, so based on using the sequence information to determine the relative flow of genetic material from our animal reservoirs, which we were sampling at the time, into the human population. So this was a paper that was published at the time. We used those methods, and we were able to come up with reasonably conclusive evidence that poultry, these three combined, and one particular poultry supply was predominantly responsible for most of the human cases that were occurring in the Manawatu, and by inference also around the rest of New Zealand as well, although we were seeing some cases attributable to ruminants as well. And this is what happened after the interventions in the poultry industry. There was a 50% reduction in human cases, an estimated huge saving to the New Zealand economy, with fewer people getting sick, fewer people looking after sick children and being off work, and the hospitalization costs were also down as well. Now, this is sustained. You can just see the Havelock North blip there. I think there was an increase in reporting happened August last year. And there is a little bit of an upturn here. We're not sure whether it's a real upturn, but that will need to be kept an eye on. So, if you remember, I mentioned that one particular strain, sequence type 474. This was the one that was responsible for most of our human cases, or the biggest proportion of our human cases, back prior to 2006. After the interventions in the poultry industry, it declined dramatically, and now we see it relatively rarely. We did some early genome sequencing studies that showed that rather than it just mutating over time, it was actually acquiring genetic material from other sources. So it was doing what we call recombination rather than mutation much more commonly. So we were interested in these questions. Why did it emerge in the poultry industry? And why did it decline subsequently? And this is where we apply our whole genome sequencing and modeling techniques. So we've gone back to our collection, and we've taken over 400 isolates going back to 2004, and we've sequenced the entire collection that we have in our laboratory. And for the first time, we can now see a lot more detail about the genomics and the population structure of this epidemic strain. I would also say this strain is very rarely found anywhere else in the world. So, and we think that most of the international cases may well be associated with New Zealand. So it seems to be a New Zealand-specific strain. And what we could see, a lot of the cases, these are human cases, so you can see these in green, and a very strong association with poultry here. And we have 3 different clades. So this is a phylogenetic tree that's kind of turned into a circle in the middle here. And you can see this clade here, which shows very little variation, this one here, which shows a bit more, and this one that's highly variable. When you add the time periods onto this plot, it shows a very interesting feature. This clade has been present throughout the entire period, so we can see ones from the early red period right the way through to the present period now, 2011. This one almost exclusively in the period 2005 to 2008, and then after the intervention in the poultry industry, we saw it jump over into another clade as well. So it's starting to evolve after the interventions happened in the industry. The other feature here was this particular gene that was present just in this group here, and there are a lot of cattle isolates. You probably can't see them very well, but there are a lot of cattle isolates in this group here, which is very unusual. Most of the isolates we saw in animals were found in the poultry population, and one particular poultry supplier. So we raised the hypothesis that maybe what happened in poultry had jumped over from ruminants, and we were interested in exploring what the evidence for that was. Now, the sequence type 474 belongs to a global group which is commonly associated with ruminants. So that was the first hint that this may have been something that crossed over from the ruminants. That gene, which I showed in the previous slide, it's an oxidoreductase gene that is also associated with ruminants in other lineages. So other lineages of Campylobacter not 474 associated also had that gene, which was a ruminant-associated gene. And the whole genome sequencing does suggest a potential jump from ruminants into poultry because of that basal group, which contains a lot of cattle isolates, then an epidemic strain, which is almost exclusively in poultry. So was this an example of host switching from ruminants to poultry, which obviously then had major consequences for public health? And so what we do here is we look at attribution of human cases. So this bar here is an attribution of all these human cases here. The ones that are not there are the animal isolates. And you can see this is the groups, the pre and post intervention period, most of these associated with one particular poultry supply, with the exception of here. And this basal group here, most of those associated with ruminants. And if we just expand that group there, what we can see is we can time the time period when this nucleid began to emerge, or the evidence from an evolutionary analysis from the ruminant source. And it suggests that, yeah, indeed, there is good evidence that this particular strain did jump over from ruminants in New Zealand into the poultry supply around about the year 2000, so just prior to it becoming a major epidemic in New Zealand. So that wasn't the end of the story. With the 474 declining, we saw a drop in the number of human cases, but then in 2014, we saw a new strain emerge in New Zealand. Now, this strain had only been found in China before, but the unusual thing about this strain was it was resistant to 2 classes of antibiotics. And this was a very big story for New Zealand at the time because we are relatively low users of antimicrobials in our livestock populations. And we have very low levels of antimicrobial resistance, generally in both livestock and in people. But these are the kinds of headlines that were seen around about Christmas time, November to Christmas, when the story broke about the emergence of this particular strain. So we first detected it in our Sentinel site in the Manawatu, and it occurred in human cases, but what was unusual was it was then found in all 3 major poultry supplies simultaneously. So unlike the previous strain, which had stayed in one supply for almost 10 years without it crossing over to any others, this one simultaneously occurred in multiple supplies. It was also associated with human cases, so we saw a big increase in the number of human cases that were resistant to antimicrobials, and also a big chicken liver outbreak that occurred in Wellington was also associated with this strain. There was no overall increase in the number of human cases. It seemed to have replaced the previous strain with this antimicrobial resistant one. So antimicrobial resistance in Campylobacter, 0 in a previous study. So the situation had changed from 0 prevalence up to around about a third of all of our isolates were now resistant to these 2 strains. So, cut a long story short, we used genome sequencing again because they were all the same strain according to our previous methods, so we had to do something more in order to get a handle on what was happening here. We wanted to ask a number of questions around the emergence, what was the basis of resistance, and how did it get so quickly into the different poultry supplies. So this is our biosecurity question. And using whole genome sequencing, we found that it was actually evolving differently in one poultry supply, the red one here, compared to the other 2. So we saw a very different pattern. So it was already rapidly evolving, even though it had probably only come into the country in 2013. It was beginning to acquire genetic material and do some very amazing things, particularly in different poultry supplies. This is work done with the Melbourne Diagnostics Unit. And most of the difference was happening in these mobile elements. So there are little things called plasmids, and in this case we actually had a plasmid, which was a circular piece of DNA, which had actually deposited both the tetracycline resistance gene and a phage in 3 different sites in the genome, and then the plasmid was lost from one of the poultry supplies but present in the other 2. So there's a lot of evolution, very rapid evolution happening, which has nothing to do with mutation, it's to do with the mobility of these genetic elements. So the kinds of key questions that we were addressing, we could now answer. The basis of the genetic resistance, we knew that tetracycline resistance was on the plasmid. There was another mutation that caused the fluoroquinolone resistance. We could date the time, because there were very few mutations, to very recently. What drove the emergence? Looking back through, it's likely that what amplified the pathogen was limited tetracycline use in breeder flocks, but not in the broiler flocks. And now we're interested using also funding from the National Science Challenge. So this is one of our projects using a combination of network analysis. So we can now look at the feed network, the bird network, the road network, and we can compare those networks with our phylogenetic network to infer what was the most likely source of transmission that was moving it so quickly between farms and between the different poultry supplies. So that's our biosecurity question. And it seems like it's most likely shared parent and possibly even grandparent stock which was responsible for the movement of a lot of these pathogens. But this is work that's ongoing at the moment. So just finally, we're using very similar techniques. So we have in combination, so this is work that we carried out with ESR. So Brent Gilpin, who's here, was also very heavily involved in leading this work. Genome sequencing in the Have a Look North incident, I think what will turn out will be there's very strong evidence for ruminants, and when you isolate an indistinguishable strain from sheep, from the wellhead, from the reticulated water supply, and from people, it's highly likely that that's what the transmission route was. We'll have to see what finally comes out, but it certainly seems the strong evidence is suggesting that that was the major route by which people got infected. And I would also point out, this is greater than 5,000 people estimated to be infected, a third of the population of Havelock North. Now, I've been involved in other waterborne outbreaks internationally and in New Zealand. This is arguably one of the largest, other than the Milwaukee outbreak of Cryptosporidium, which had over 400,000 people affected. This is one of the largest waterborne outbreaks ever in the world that's ever been recorded. So it's quite remarkable what happened there. So just to conclude, using the new tools and technologies, and this is not just the genome sequencing technology, it's combining it with better models which use our understanding of evolution and transmission. We can do things such as estimate incursion dates, so when pathogens came into the country, what happened to them subsequently, how they became transmitted around livestock populations and between people. We can use it to determine what the sources of human infection are and use that to put interventions in place to try and reduce the incidence of human infection. We're interested in transmission between food sectors. I mentioned the cattle and poultry story, and between poultry companies, and we're also looking at other pathogens with the challenge as well, such as BVD and Staph aureus, and how those are transmitted between livestock populations. We're combining information from pathogen sequencing with evolutionary modeling and network analysis. You do need to have isolates from multiple sources to make inference, and this is something we need to be mindful of, is we do need to have isolate collections which are sequenced and available in order to make these kinds of inference. And collaboration, data sharing, absolutely essential. Just loads of people involved in the work that I've described there. The team, MEPILAB team there, fantastic group of people, a very multidisciplinary team in the Hopkirk Institute. Carolyn and Sabrina. Sabrina's the PhD student that's funded by the Challenge, working on the network analysis. And we also have a very big thanks, obviously, to our main collaborators at ESR, and particularly to the many, many people at ESR, as well as all these other people which I can't mention individually. Thank you.
Thanks, Nigel. We've got time for a couple of questions.
Barry.
Thanks, Nigel. Good talk and good science. Transmission from ruminants to poultry of Campylobacter, is that through blood and bone meal, do you think? And if so, did you sample sparrows? And has there been transmission into sparrows and potentially into other wild bird populations?
We haven't isolated that particular strain from any wildlife source, but we have isolated it from environmental water and from ruminants, mainly cattle. I think it's unlikely that it's feed, given how fragile Campylobacter is. I think it's most likely it may well have been environmental cross-contamination that happened. And we know that there's a lot of poultry farms in particular that are neighbouring a lot of cattle areas. There's a lot of poultry farms in and around the Waikato, for example. So I don't think we'll ever know exactly what happened, but what we can infer is that historically there's good evidence that it did cross over from the ruminant supply into poultry.
You couldn't whisper who poultry supplier A might be?
No, I can't.
Damn.
No, I think I would say that the poultry industry have been extremely helpful and funded work on genome sequencing, which I think is amazing for an industry, and they did really get together. I think there's still a lot that could be done, but I don't think it's helpful to name individual suppliers. Yeah.
Okay, thanks.
Okay, thank you.